Once again, good afternoon to one and all. We will start our practical class in continuation to previous one, that is about introduction to biochemical test. Now, this is about the secondary biochemical test. So, secondary biochemical test, the objective of today's class, that is about principle, procedure and interpretation of various biochemical tests. They are considered as secondary biochemical tests, and all this will again help for classification, identification of various bacteria up to genus and species level. So last class what we had studied. So last class, if we can recapitulate that what we had studied, then we had discussed about primary identification of bacteria. So inside the primary identification, we had also dealt with the primary biochemical test. So along with primary biochemical test, there are other identification method that help us to identify and classify the bacteria. For example, gram staining. The alternative of gram staining is Lana test and Keyword test. Then growth on Mekong-Ki agar, that will help to differentiate gram positive from gram negative, that contains some specific agent like bile salt that will promote the growth of gram negative, whereas crystal violet that will inhibit the growth of gram positive bacteria. And then we had also discussed about pair of these tests, catalase and oxidase. This is also important to classify bacteria up to some genus. Okay. Then motility test, we had discussed in depth about various tests by which we can assess whether bacteria are motile or not. We had also discussed in brief about OF test. Okay. And we had seen that all the biochemical tests are generally based on this particular principle, that bacteria possess some enzyme. We have to demonstrate that presence of particular enzyme by providing the substrate. So that substrate will be cleaved by this enzyme and that will produce some product. That product might be directly visible to your eye, or that requires some addition to give a visible result. So this is what we had studied till now. Now we will move to the next, that is today's class. We will discuss about this biochemical test. These are not the sole secondary biochemical tests. There are others also that I had given as a part of your assignment. But in depth, we will discuss about TSI test, IMViC test, urease, phenylalanine DA-, and at the end, we will also discuss about nitrate reductase test. So these are the basic important tests that helps to classify or to identify some species of the bacteria. Now, we will see the first test, that is about triple sugar iron test. So triple sugar iron test, generally name itself indicating something. Triple sugar means it contains the 3 sugar, and iron means it contains some ferrous component that helps to identify something. So we will see here that generally in the some laboratory they are using some alternative media to TSI. So alternative media, that is also known as Klinger's iron agar. This is similar to TSI agar, but nowadays this is not used commonly, and people are generally use TSI agar, and you will get a lot of information publication with TSI. So TSI agar that contain 2 important things. One, that contain the sugar, and second, that contain the source of iron. So generally triple sugar means that contain 3 sugar, namely glucose, lactose and sucrose. Whereas this ferrous or iron source, this iron source will help to identify whether they are producing hydrogen sulfide or not. And to see these changes, the media contain phenol red. So phenol red is a pH indicator that will tell us if it is alkaline, then it is red color. If there is acidic, then it will convert it to acidic, that is yellow color. Now in general, this TSI test is used to differentiate Enterobacteriaceae, and in specific this will help to identify the Salmonella species. The principle we had discussed earlier, and here also you can see that sugar utilization and reduction of sulfur to hydrogen sulfide, these 2 things we are targeting, and we are observing this two phenomena by change in pH. Now coming to the procedure, that how we can do this test. So generally TSI agar we have to take, and this TSI agar we have to stab inoculate your culture. Okay, so after inoculation, stab, or we can also do the strict culture. So stab and strict culture you have to do, then you have to go for incubation. In general, overnight incubation is sufficient, and after getting sufficient growth, you can go for performing this test. So on the next day of inoculation, we can observe this type of changes. Okay, so this particular tube that shows that now before coming to this one, you should know that when we are preparing the slant, okay, so this is considered as slant and this portion is considered as a butt. Okay, so butt and slant, so here color change with respect to slant and butt is indicative of result. So red slant and red butt. Okay, this is a normal uninoculated or the organism which are not fermentative, they are considered or they are showing the red butt and red slant. Now, if you will see the dark or black color, okay, dark and black color, that is indicative of production of hydrogen sulfide. So such bacteria are producing hydrogen sulfide and that's why they are positive for H2S production. Now, if there is a third combination where your slant is a red but your butt is yellow, so this indicates that only glucose is fermented and other two sugars are not fermented. So this is because of some fermentation, this other two may not be fermented. Now the fourth combination that is possible, that yellow slant and yellow butt. So you can see the slant is also yellow, butt is also yellow. So both thing is yellow, that indicate that all the sugar, lactose and or sucrose with glucose, they are fermented and they produce the acid. That's why it changes color from normal red to yellow one. So this is the interpretation of TSI. You can observe such changes on the next day. Here I give for example for salmonella, we are writing this observation or result like this, that red tell that slant is red, white is indicate that your butt is yellow, and plus H2S plus indicate that salmonella produce hydrogen sulfide. So likewise other organism also they have that typical result or combination of this result. So this is in brief about TSI. Now we are moving to the battery of tests, that is abbreviated as IMViC test. IMViC test is most important with respect to Enterobacteriaceae family. So IMViC test, generally this is an abbreviation because we are performed 4 tests and this is indicated like this. Okay, so indole, methyl red, VP and citrate. So first letter that will form a IMViC. This is abbreviation. And generally this is used to differentiate various Enterobacteriaceae groups. Now we will see one by one test and we will see the important result and important principle of this test. So first we will start with the indole test. So generally indole, so there are some bacteria which can produce the indole. So what will happen? The tryptophan, that is an amino acid, this tryptophan, when bacteria is capable to utilize this tryptophan, they will convert it into indole along with other pyruvic acid and ammonia. So generally this compound, pyruvic acid and other compound, they are required to synthesize some cellular component and that's why it is important for their survival also. So this is a characteristic of some bacteria, they can utilize tryptophan and produce the indole. So if we can demonstrate the presence of indole, then we can say that particular group of bacteria contain this tryptophanase enzyme. Now the procedure is again very simple. You have to take the trypton water tips. This is a broth. In this broth you have to inoculate your test bacteria. Whenever you are inoculating test bacteria, you have to always keep negative control, positive control and uninoculated. Uninoculated means you need not to inoculate any bacteria. That will tell us that it is okay, your media is okay or your media get contaminated. So always you have to put at least 3 controls along with your test sample. So after inoculation you have to go for incubation. In general we are performing overnight incubation that is 14 to 16 hours overnight at 37 degrees centigrade. On the next day, you have to add the coax reagent. So coax reagent, when you will add from the side, approximately 10 or 12 drops, if indole is produced, then that will produce the cherry red color ring. So cherry red color ring on the top, on the addition of coax reagent, on overnight incubation of bacteria in tryptone water, that will indicate that it is positive for indole test. And this, if there is a no ring or colorless, or you can say yellow, that will tell that indole test is negative. Now second test, that is known as methyl red test. It is also abbreviated as MR test. So MR test, generally it is used to determine the ability of bacteria to oxidize the glucose. And once glucose is oxidized, then there might be possibility of production of two products. One, that is stable in acid, that is acid production, and second product, that is neutral. Okay. So these two, if acid production or methyl red, if acid is produced, then we can say that they are MR positive. If there is a neutral product, then we can say that VP positive. VP, that is a next test. So general to perform this methyl red and VP test, a common broth is used, that is abbreviated as MRVP broth. This is abbreviation of methyl red Waugis Prosper broth. And this broth contain GPP water. GPP means glucose phosphate peptone water. So glucose is act as a substrate, and along with this, methyl red is added as a pH indicator. So pH indicator, what will happen at the low temperature, at the low pH, this methyl red is red in color, and as the pH is increasing, or you can say that is going towards alkaline side, that will convert it to yellow color. So color change is always depend on what type of pH you are using, or what type of pH is added in your broth or your in test media. So always you have to remember that what type of pH indicator is added. So here methyl red is added as a pH indicator. Now we can see how we can perform this methyl red test. Again, you have to take some tubes, required tubes. You have to inoculate the test bacteria along with positive control, negative control, and uninoculated. As mentioned earlier, you have to incubate for overnight, and then on the next day you have to add the 5 drops of methyl red indicator. So this methyl red indicator, when you are adding the methyl red indicator inside overnight incubated culture, if that will produce the red color, this red color indicate that glucose has been oxidized, and that will show that MR is positive or MR test is positive. So this is about the methyl red test. Now a similar or combination of MR, that is VP test. So VP test is also performed on the same broth. So whatever overnight incubated broth is there, you have to divide in 2 test tubes, and you can perform MR and VP test simultaneously. Here in VP test, we are targeting non-acidic or you can say neutral end product. So if there is a presence of neutral end product, if we can demonstrate, then we can say that VP test is positive. So here what we have to add on the next day we have to add 2 reagent. First, that is Barrett's reagent A. Generally this Barrett's reagent A contain alpha naphthanol, and followed by addition of Barrett's reagent B that contain 40% KOH. So once we will add these 2 reagent in a sequence of reagent A followed by reagent B on the overnight incubated your test bacteria, and you can mix it for 10 to 15 minutes, you will found a rose color. So if rose color is formed on the top or in the tube, then you can consider that it is a VP positive. If there is no change in color, there is a no rose color is produced, then you can say that VP is negative. So VP and MR both are the almost similar test, only we are demonstrating that end product is what, and based on that we are adding the different reagent. Now coming to the last in IMViC test, that is a citrate test, and this is abbreviated as C in IMViC. So citrate, generally what happens, many bacteria contain citrase enzyme. So this citrase enzyme that will help to reduce the citrate to pyruvic acid, acetic acid, and some other products. So now what we are doing, we are providing specific media that is known as Simon citrate agar, and this Simon citrate agar contains citrase as a sole carbon source. So that agar contains the citrate as a sole source of carbon, means that bacteria has to utilize the citrate, and bacteria can utilize citrate if they possess the citrase enzyme. Okay. So now what happened? We have to take the tube citrate agar slant. That citrate agar slant, earlier I had told that contains the citrate as a sole carbon source. Here the pH indicator is bromothiamol blue. So bromothiamol blue, that is a pH indicator present in Simon citrate agar, and when you are preparing the media, is normally green in color. So after preparing this media, you have to go for inoculation by streaking, incubate, and observe for color change. So if test bacteria are capable to utilize the citrate as a carbon source, and they possess the citrase enzyme, then that will produce the Persian blue color. So this is a Persian blue color. So normally green color is present as a Simon citrate agar, and if they are utilized by the citrate, or bacteria are utilized the citrate, then they will convert it to Persian blue color. So presence of Persian blue color, that is indicate taste is positive, and no change in color, that indicate taste is negative. Generally E. coli is citrate negative, whereas Salmonella is citrate positive. This is just summary of IMViC test, that how we can perform, what media we are using, what substrate we are using, what reagent we have to add, and how the positive and negative result is observed. So this is just diagrammatic representation that will help you to remember for your exam. So this is in brief about IMViC, and here I would also like to highlight that you should remember all the IMViC pattern of Enterobacteriaceae bacteria, like E. coli, Salmonella, Proteus. Okay, you should remember, because many times in exam fill in the blanks or multiple choice they are asked. So they will simply write, give the IMViC pattern of Salmonella. Then you should know. Okay, even though it is covered in your theory, but you should remember, or you should find out as a part of your assignment of today's class. Now moving to the next next test, that is urease test. Okay, urease test generally in first class also I had mentioned that anything that is prefix by ASE, suffix as ASE, that is indicate enzyme. So urease, here urease is an enzyme that we are targeting. If any bacteria that contain the urease as enzyme, they will utilize urea, and that will produce some product. And generally urea is degraded, or they will produce the ammonia that will produce the ammonia, and that we can detect as a indicator. So general in urease test we are using phenol red as a pH indicator. So phenol red is already added in your agar, and once urea is utilized by your test bacteria, then that will help to demonstrate by changing the color. So the procedure is again simple. You have to take the urea broth, you have to inoculate your test bacteria, incubate at appropriate time and temperature, and on the next day you have to see the color change. Alkaline end product is produced, then phenol red will turn yellowish to pink. So you can say pink is a positive for urea test, whereas yellow color is negative in urea test. So this is about urease test. Now moving to the second last test, that is phenylalanine deaminase test. Here also this is a name of enzyme. Now this phenylalanine deaminase, this enzyme that is particularly useful to degrade phenylalanine into phenyl pyruvic acid and ammonia. So this is again we are targeting by pH indicator or by adding some reagent that will help to demonstrate the presence of end product. So what we are doing here, we are taking the 2-phenylalanine agar slant, we are inoculating our test bacteria along with appropriate controls. We have to go for incubation to grow them in that tube, and on the next day, this is not produced directly color change. So what we have to do, because phenyl pyruvic acid, what is the end product of that breakdown, that is colorless. We cannot see that particular product. So what we have to do, on the next day we have to add 10% ferric chloride. This acts as an oxidizing agent, and once this 10% ferric chloride is added and phenyl pyruvic acid is present, then that will change color from yellow to green. So yellow to green on addition of 10% ferric chloride on overnight grown bacteria, that will indicate that phenylalanine deaminase test is positive. So this is something different from previous test, where end product is colorless, we cannot observe this end product, and we require to add something, we require to add some oxidizing agent so that we can easily observe these changes. So this is leads to green coloration, and green color is positive in case of phenylalanine deaminase test. Okay, green color is negative in case of citrate. Okay, that Persian blue, that is positive. So this will create the confusion. So you have to remember along with your pH and pH indicator and the color of positive test and color of negative test. Now, moving to the next test, that is nitrate reductase test. So nitrate reductase test is the last test of our class, and this is one of important test because their observation is little bit complicated. So previously what we had seen, we are just inoculating that particular test bacteria and we are going for observation of color change on the next day. Then we had seen that there is one test where we have to add the additional oxidizing agent to observe the color change. Now here we require 2 or 3 steps or 2 or 3 reagents to get the final concluding result of nitrate reductase test. So nitrate reductase test, what basic principle behind this one is, there are some bacteria that can reduce nitrate to nitrite. So those bacteria, they have enzyme like nitrate reductase, they can reduce nitrate to nitrite, and this reduction can be easily seen by formation of red color on addition of some solution. So this is a basic principle. Now we can say how we can perform this test. So we have to take the nitrate brood tubes. Again, that is number of tubes depend on your number of samples. You have to inoculate your test bacteria along with positive negative controls and uninoculated. Go for incubation overnight, and on the next day you have to perform this test. So what you have to do, on the next day you have to add the solution A. Solution A contains generally sulfonilic acid, and that will be followed by addition of solution B in that particular broth. That solution B in principle contain alpha naphthylamine. After addition of these 2 solutions, you have to observe color change. So after addition of solution A and B, if you will observe red color, then you can say that nitrate test is positive. This is clear to all. Okay, overnight culture, next day you have to add solution A followed by solution B, and you have to see if there is a red color, you can say nitrate reductase is positive. Now, little bit complication is that now no color change. For example, okay, red color, that indicate undoubtedly that nitrate test is positive. But there is a scenario where there is no color change on addition of solution A and B. Then there might be 2 possibilities. One possibility is that nitrate were not reduced. So nitrate is present as such in that particular broth. And second possibility is that there are some organisms, they possess some potent nitrate reductase enzyme. And because of this potent nitrate reductase enzyme, they can reduce nitrate beyond nitrite, like nitrogen gas and ammonia, and that is difficult to demonstrate in this particular step. So now these two possibilities we have to evaluate whether they are true positive or true negative. So what we are doing in subsequent step, that we are adding solution C. Solution C is generally zinc dust. Okay, so we are adding the pinch of zinc dust to that particular broth in which we had already added nitrate A and solution A and solution B, but we could not observe the red color. So that negative, we are adding the solution C in that one, and on addition of zinc, if red color is formed, then that will tells that the nitrate taste is negative, because whatever nitrate is present, that is reduced by zinc, and now they are giving the red color. So this is cross checking whether nitrate is present or not. Okay. Now the second scenario is, even if you are adding the zinc and there is no production or no production of color, that will indicate that your original broth does not contain nitrate. Nitrate is already reduced to nitrite or beyond to nitrite, like ammonia and nitrogen gas. So that's why nitrate is not present, and even after addition of zinc, that cannot reduce to this particular product, and that's why it will not give color. And such cases, we are consider them as a positive reaction. So this you have to remember. Now this, I have tried to simplify this observation by putting this slide. So now we can see that solution A and solution B we had added in three different test bacteria. I had wrote name over this one. Okay. Now on addition of solution A and B, if we are getting the red color, that undoubtedly indicate that this particular bacteria is reductase positive, nitrate reductase positive. There is no doubt. Now the doubt is between this tube and this tube, where there is no color production, or you can say they are the unreactive tubes. So what we are doing this, in this unreactive tubes, we are adding solution C. Solution C, that is a zinc dust. So a pinch of zinc dust is added. If that will produce the color, for example, this is Corynebacterium xerosis. So if this produce the color, that will tells that nitrate is present and taste is negative, or organism is negative. But there are some extreme cases like Pseudomonas aeruginosa. They have the strong reductase enzyme, and because of strong and potent reductase enzyme, what will happen? This nitrate is reduced to beyond nitrite, and that will produce nitrogen ammonia like this. So that's why there is no presence of this one, that we are trying to assess by our solution A and B. That's why this indicate that even though there is a no color production, this taste is considered as a positive. So this is in brief about the important secondary biochemical test, and this is the part of your assignment. I had enlisted the two thing and important thing you have to remember from my side along with all listed assignment, that you should prepare one list of pH indicator. What type of color they are giving in acidic pH and what type of color they are giving in alkaline condition. So there are 3 or 4 or maximum 5 pH indicator generally used. So that table you have to prepare and you have to share. And second important, that is a IMViC pattern of enterobacteriaceae. This is I had highlighted because this may ask in your exam too. That's why you have to find out and you have to remember the IMViC pattern of important enterobacteriaceae family. I thanks all of you for listening me, and if you have any query, you can divert me on this number or my email ID. I am happy to answer you. Thank you one and all for listening me.